Okay, good morning, part two. We'll get started. For those of you who haven't met me, which will probably be the biological scientists amongst you, I'm Patrick Lewis. I'm an associate professor in the School of Pharmacy. And today I'll be talking to you as an introductory lecture, really, on the concept of enzymes as drug targets. As for all of my lectures, I'll be screencasting this, so there will be a video available on YouTube for you to have a look through at a later date. In terms of the lecture objectives, learning outcomes, however you want to describe it, at the end of the lecture you should be able to discuss how enzymes can be drug targets and actually why they're so important as drug targets, and list examples of enzyme inhibitors in clinical use as drugs. You should be able to define the kinetic parameters of enzyme catalyzed reactions, in particular looking at the concept of Km and Vmax, and the different effects of different types of enzyme inhibitors on these properties. You should also be able to explain the meaning of Ki, a constant measuring inhibition as a measure of the potency of an enzyme inhibitor. Just give you, yes? I've just said I'm screencasting it, so there'll be a video of this. Yeah. In terms of what I'll be talking to you, just to give you an overview of what I'll be covering, I'm going to go back to what is hopefully familiar to you from part one and talk a little bit about enzymes as biological catalysts. So what is the nature of the target that drugs are trying to hit? Talk a little bit about enzyme kinetics. Again, this should be a refresher from last year. If it isn't, what I would strongly recommend is going back and looking at some of your lecture notes from last year, maybe even accessing material. There's a lot of material on the internet explaining enzyme kinetics. And for the second half of the lecture, what I'll do is I'll talk through a couple of examples of enzymes as drug targets from various areas of clinical medicine and drug discovery, and talk a little bit more about the mechanisms of drug action. So, as I said, this should be a recap for all of you. Enzymes are biological catalysts. They make reactions happen with greater ease, with lower energy, and with greater rate. They're basically protein machines. They're biological catalysts. They're made up of amino acids assembled into three-dimensional structures that carry out tasks both within cells and outside of cells as well. And critically, they're important and central to all aspects of cellular function, both bacterial and eukaryotic. And that's one of the key reasons why enzymes are such important drug targets for clinical medicine, and actually more widely in terms of biotechnology. Enzymes are a key component of biotechnology development. And to give you an idea as to exactly how important the enzymes are as drug targets, here's an overview of current drug targets. So for licensed drugs that are used in clinical practice, this is from a while ago, back in 2002, but this is still very much the case today. And if you look across all of the different targets that small molecule drugs are hitting, actually almost half of them are enzymes. And there's a variety of other targets which are approached by drugs in other concepts. But enzymes represent the largest single group of molecular targets in cells for drugs that are in clinical practice, that are FDA approved or approved by the European Medicines Agency. And really, that highlights why this is important, both in terms of pharmacy, in terms of drugs that may be prescribed going into clinical practice, but also in terms of understanding drugs in the broader arena of biology. So, what are enzymes? They're biological machines, they're biological catalysts. And they are the three-dimensional organization of an enzyme allows them to have specialized binding sites that are specific for certain substrates. The basic function of an enzyme is to bind that substrate and to modify it. And this binding pocket is the active site. And this is a cartoon providing a very simplistic overview of the fundamentals of an enzyme operating. So you have the three-dimensional organization, the three-dimensional conformation of a protein enzyme, and you have a substrate that enters the active site of that enzyme. The three-dimensional organization of the active site will be specific for that substrate. The enzyme changes shape slightly upon the binding of the substrate and may form non-covalent links with the substrate that stress the internal organization of bonds within that substrate. There is then a chemical reaction which results in the modification of that substrate, and products leave the active site. So that's a simplistic overview of how an enzyme works. Looking at this energetically, basically what enzymes do is they reduce the activation energy of a chemical reaction. So what this boils down to fundamentally actually is a chemical reaction. So here you have the reactants, in this case using the example of CO2 and water. Now without enzymes, a reaction to combine CO2 and water has a high activation energy, and there's an activation energy that needs to be overcome in order for this reaction to occur. Now, in the presence of a catalyst, and that can be an inorganic catalyst as well, there are, of course, inorganic catalysts which can accelerate or lower the activation energy of a chemical reaction like this. But an enzyme acts in the same way to reduce the activation energy to release the products. And really the key point about what an enzyme does is it reduces that activation energy. It makes the reaction more likely to occur. And if you think about all of the processes that occur within your cells, whether that's generating energy from sugars through the respiratory chain, whether that's breaking down waste products, whether that's generating new proteins, new amino acids, all of these require chemical reactions combining compounds within cells. And so without enzymes to reduce the activation energy of those reactions, your cells simply would not function. In terms of the kinetics underlying all of this, so chemical description of the way in which enzyme reactions occur, you can measure this in a number of different ways. And traditionally, the way in which to do this is to look at the substrate concentration and the initial rate of the reaction. You can follow this in vitro, so you can follow this in a tube, put a substrate in, put your enzyme in, and follow the rate of reaction. For certain reactions, actually, you can follow this very easily using a colorimetric output. This allows you to define a number of parameters for the reaction. Two of them are key concepts that you need to understand in order to understand how we target enzymes as drug targets. This is Vmax, which is the maximum rate at which an enzyme reaction can occur at the highest substrate concentration, and the Km, the substrate concentration at which the enzyme will work at half Vmax, and the Km is defined as half Vmax. And these can be defined experimentally in vitro. In terms of an example of how you can actually measure this in vitro, for those of you in pharmacy, last year we carried out a series of experiments measuring the activity of an enzyme called lactase. Lactase breaks down lactose, which is found in milk, into degalactose and deglucose, and catalyzes this step here. By adding in water, you break apart the bond between the two sugars, release these substrates. This is actually very important in terms of nutrition. Our bodies can metabolize galactose and glucose, they can't metabolize lactose, so you need to have an enzyme that breaks these apart. And actually you can measure this in vitro using a simple colorimetric measure causing the release of orthonitrophenol. For those of you in SBS, there's a YouTube video of this practical, and there's also a guide to the and background to this online, which I'll circulate after this lecture. The key point here is actually you can measure how fast the reaction occurs by a change of your substrate from colorless to a yellow color. So you can actually define experimentally the rate of the reaction. And this allows you to generate data points like this. So you increase the substrate concentration and measure the initial reaction rate. That allows you to define the Vmax and the Km. And these are key concepts. If you don't understand Vmax and Km, go back, look at your notes from last year, or have a look through the material which is on the internet. There are a number of ways in which you can present this data or project this data graphically. This is a Lineweaver-Burke plot, which is a very, very useful tool in terms of understanding how Vmax and Km alter as you alter the conditions which an enzyme reaction occurs in. This is the reciprocal of the substrate concentration and the initial velocity. So it's basically taking these data points and plotting them as reciprocals. And what you get is, instead of that, the curve going up the graph like that, you get a linear line, a linear plot of those data points, which you can then fit and extrapolate to the x and y axes. This allows you to calculate quite accurately both the Vmax, which is the point at which the best fit line crosses over the y-axis, and the Km, which is the point at which the plot crosses over the x-axis. And if you invert the reciprocal for those points, what you can calculate is the Km and Vmax. Now, as I say, these are critical in terms of understanding how we target enzymes in clinical medicine. And the reason is that what we're doing when we expose enzymes to manipulation, to inhibitors, is we're actually altering those enzyme kinetics. We're modifying the way in which that reaction works. So here again, we have a simple cartoon showing how an enzyme works. This is in the normal situation where you have the substrate binds to the enzyme into the active site. The enzyme binds it, the enzyme changes shape, catalyzes a chemical reaction, releasing the products. And essentially what we're doing in terms of targeting enzymes in drug discovery is we're looking to block or modify that process. And there are a number of ways in which we can do this. At a fundamental level, what we're doing is we're generating small molecules which can act to bind that active site and modify the function of the enzyme. I'll talk a little bit at the end at other ways in which you can target enzymes, but the vast majority of drugs in clinical practice are drugs that directly target the active site. Most of them do so by mimicking the three-dimensional shape of the substrate. Now, if you have an inhibitor that binds to the enzyme and binds to that active site, what it does is it can compete with the substrate to prevent the substrate binding. So if you think of this as a chemical reaction, you have the enzyme, you have the inhibitor, you have the substrate. If you have more of the inhibitor around and it has the same binding properties to the active site, then the inhibitor, if it's available in a molar excess compared to the substrate, will bind the active site more than the substrate, and so less of the substrate will be processed and changed into the products. And that's the way in which a competitive inhibitor works. Basically competes directly with the normal substrate of an enzyme to prevent that substrate being processed. And you can represent this in terms of a series of binding products and constants. So here you have the enzyme and substrate. If you add in an inhibitor to this equation, you can then form an enzyme inhibitor complex. And here you have one of the inhibitor constants that allows you to define the binding properties of that inhibitor to the enzyme, allows you to define one aspect of competitive inhibition. It can also act at the point where a substrate is bound. So actually an inhibitor can also bind to the active site at the same time as the substrate, thereby preventing the substrate being modified, giving you a second potential inhibitor constant. And what both of these do is they reduce the possibility of an enzyme catalyzing the reaction that it's meant to and releasing the products. And you can actually view this in terms of the enzyme kinetics as well. So up till now, I've talked about competitive inhibitors. So these bind non-covalently to the active site of an enzyme and modify the likelihood that a substrate will bind to that active site. So here's the Lineweaver-Burke plot that I showed you a couple of slides ago, showing the reciprocals of the substrate concentration and the initial velocity. Now, if you put in a competitive inhibitor, what you do is you alter the gradient of that plot, and it shifts to a steeper gradient, as you can see here. Now, the consequence of that is quite important, because you can see that actually the point at which this crosses the y-axis, the point at which the Vmax is determined, actually doesn't change at all. What changes is the point at which it crosses the x-axis. And this really gives you an idea as to exactly what a competitive inhibitor will do in terms of enzyme kinetics. Because if you alter the point at which it crosses the x-axis, what you're doing is you're altering the Km, the substrate concentration at which the velocity of the enzyme reaction is at half of the Vmax. In terms of actually the the practical consequences of that, you need more of the substrate in order to get the products. So by putting in an inhibitor, you're reducing the likelihood that a substrate will be processed into products. Up till now, I've talked about competitive inhibition. There is also a key further class of inhibition in terms of targeting enzymes, and this is non-competitive inhibition. And in this case, instead of binding to the active site and competing directly with the substrate, a compound, a small molecule, a drug, will bind to the active site and covalently modify the enzyme. By doing so, what it does is it becomes part of that enzyme and actually cannot be removed from the active site. And clearly that has big consequences in terms of how an enzyme will function. Because if it has an inhibitor bound to the active site, occupying the active site covalently in a way that can't be removed, what that means is that it can't bind the substrate at all. So there won't be an exchange of the inhibitor and the substrate into the active site. This actually has a slightly different consequence in terms of the enzyme kinetics. So if you look down here at the bottom, you can see the consequence of a non-competitive inhibitor, one that covalently modifies the enzyme complex. And here, actually, you don't modify the Km. So you can see that the point at which the best fit line crosses over the x-axis actually doesn't change. So the Km is unaltered. What is altered is the Vmax. The point at which the line crosses over the y-axis. So this has a major impact on the function of the enzyme. And basically, for any enzyme which is bound by a non-competitive inhibitor, actually you remove that from the pool of enzymes capable of carrying out a task. A competitive inhibitor can actually be removed from the active site, can be washed out, and eventually its impact will be reduced. So what I'll do, that gives you a bit of background in terms of the way in which we can target enzymes, in terms of the theoretical background, in terms of the impact on enzyme kinetics. It gives you an introduction to some of the classes of inhibitor that are used in clinical practice. What I'll do over the next 20 minutes or so is talk through examples of how this actually works. So examples of compounds that are approved for human use, which target enzymes in clinical medicine. The key point, the first couple of examples that I'll use are enzymes targeted in infectious agents, and in particular in bacteria or viruses. So bacteria and viruses, just like all of the cells in our body, require enzymes to function. Bacteria have a set of enzymes, some of which are very similar to the enzymes found in our cells, some of which are quite different. Without those functioning properly, bacteria cannot replicate and can be killed. Viruses equally, although they're not cells, many of them require the activity of specific enzymes, some of which they encode themselves. And without the function of these enzymes, actually they cannot replicate. They cannot go on to infect other cells, and they cannot cause damage to infected cells. And one of the important concepts in terms of targeting these infectious agents is that there are enzymes that are found in bacteria and in viruses which are not found in human cells. And this gives you a great opportunity, because what it means is that you can get differential effects. You can target enzymes in a bacterium that you can't target in a human cell because the enzymes simply are not there. And what that does is it allows you to actually have a bactericidal effect. You can kill the bacteria, prevent them from replicating, or completely get rid of them. And the majority of drugs that are used as antibiotics to target bacterial replication and bacterial infection are drugs that target bacterial enzymes that are not found in the human proteome. And that gives you differential toxicity. It means you're toxic to the bacteria, you're not toxic to the human cells. Equally, viruses have a wide range of enzymes, some of which, again, are not found in the human proteome. And by specifically targeting those viral enzymes, you can prevent the viruses replicating or get rid of infection altogether. And I'll talk through an example of both. In terms of targeting bacterial enzymes, probably the best studied and the most widespread drug in use are drugs targeting DD transpeptidases. And this is the DD transpeptidases are a class of enzyme that act to cross-link the peptidoglycan wall subunits for bacteria. So actually constructing the bacterial cell wall that sits outside of the membrane. These proteins are also known as penicillin binding proteins or PBPs for reasons that will become very clear. So what the enzyme does, going back to the simplified cartoons I have, what this is actually exactly the same, but with a specific example. So what you have is the peptidoglycan wall, you have the DD transpeptidase, it binds to the wall, and carries out a cross-linking reaction, allowing the bacteria to expand its cell wall and provide protection to the bacteria from the outside world. Once it's carried out its job, the DD transpeptidase will release the covalently linked wall and move off and carry out another task. And what beta-lactam antibiotics, the first of which was that was identified was penicillin, what they do is they bind to and irreversibly inhibit DD transpeptidases. So they go into the active site of the DD transpeptidase, they form a covalent bond with the active site, and they act as a non-competitive inhibitor of that reaction. And that has quite serious consequences for the bacterium. This is penicillin, and the beta-lactam ring, which is this ring just here, is almost like a sort of a trap that's ready to spring for the bacterium. This binds to the active site of the DD transpeptidase, and one of the bonds within the beta-lactam ring forms a covalent bond with the active site of the DD transpeptidase. What that does is it inhibits the action of the transpeptidases, and it prevents the bacterium building new bacterial cell walls. Now, that in turn actually has very serious consequences for the bacterium, because if a bacterium, which is always trying to replicate and grow, cannot build new cell wall, eventually it will pop out and will lyse. And this is how the penicillin and the beta-lactams cause bacterial cell death. They cause the bacteria to come out of its protective cell wall and lyse. And actually, we understand this in a great deal of detail. Clearly, the beta-lactams are one of the most important drugs in clinical use, given the impact that they've had on infectious diseases driven by bacterial infection. And so there's been a huge interest in really understanding this. What you can see here is actually a crystal structure for a DD transpeptidase. So this is the protein structure surrounding the active site. And here in the active site, you can see a beta-lactam antibiotic bound to that active site. So we actually understand this in exquisite detail. We have angstrom resolution level images of what a beta-lactam antibiotic looks like when it's bound to a DD transpeptidase. Now, of course, as you'll be well aware, actually bacteria have their own defense mechanisms against this, and this also relies upon enzymes. So the beta-lactam ring, which is shown here, is the key chemistry that allows a beta-lactam antibiotic to target a DD transpeptidase. And bacteria have evolved over many millions of years to be able to resist and break down those beta-lactam chemicals. And so a number of bacteria express beta-lactamases, enzymes which bind and hydrolyze the beta-lactam ring, inactivating it and preventing it from targeting DD transpeptidases, therefore allowing a bacteria to grow as normal. And actually, this is one of the key mechanisms whereby bacteria develop antibiotic resistance. So if you read in the press about MRSA or methicillin-resistant Staphylococcus aureus, the reason Staphylococcus aureus is able to be resistant to methicillin is the production of a beta-lactamase enzyme. That gets rid of the beta-lactam antibiotic and prevents it from doing its job, blocking cell wall manufacture and killing the bacterium. So in a way, this is a kind of enzymatic chemical warfare that goes on between the bacterium and the medics trying to treat bacterial infection. And what has gone on over the past 50 or 60 years as MRSA and other forms of beta-lactam resistant forms of bacteria have become a greater and greater medical problem is that we've been able to develop beta-lactams with a more complicated structure that are resistant to that hydrolysis by beta-lactamases. As time has gone by, actually, the bacteria have fought back. Their own beta-lactamases have evolved and been able to cleave these new substrates. And this is basically how bacterial resistance to antibiotics proceeds. This is using the example of beta-lactamases and DD transpeptidases. But actually, this is true for almost all forms of bacterial drug resistance. The second example I'll talk through is an enzyme which is found within the human immunodeficiency virus, the virus which causes immune dysfunction, eventually resulting in the development of AIDS as a clinical syndrome. HIV is a retrovirus. It's an RNA virus, so if you looked at the structure of the HIV virus itself, it contains RNA within a capsid shell. And there are a number of enzymes which are absolutely required for HIV to replicate and to infect cells. One of these is reverse transcriptase. So as an RNA virus, what HIV does is it copies its RNA into DNA and inserts that into the host cell genome. So it actually integrates with the DNA of the host cell. That makes it quite tricky to target HIV once it's carried out an infection, because it actually becomes part of your cells. And this is true for all retroviruses, and there are a number which are clinically important, including HIV. Importantly, though, there are a number of enzymes that are present in retroviruses and in other viruses which are not present in the human genome. Reverse transcriptase is one of these, but there's an additional HIV protease, a protease being a protein which cleaves other proteins, which is present within the genome of HIV, but is not present in the human genome. The HIV protease is central to HIV replication. If it isn't able to cleave the proteins that are produced from its own genome once it's integrated into the host cell, it cannot replicate and cannot produce additional viral capsids. And so by targeting this protease, actually you can stop HIV replicating, or you can certainly slow down and reduce the level of replication. This is exactly how one of the key clinical drugs targeting HIV and in use clinically to reduce and to slow down HIV infection works. This is atazanavir, which is produced by Novartis, the chemical structure of which is shown here. What this does is it binds to the HIV protease, and again we have a crystal structure for the HIV protease. It binds to the active site as a competitive inhibitor and prevents the protease cleaving the viral proteins to produce new capsids. And if that's prevented, actually, although it won't destroy the virus, it won't prevent the virus infecting the cells that it's already in, what it does do is it reduces the ability of the virus to produce new viral capsids and infect other cells. So it reduces the spread and the activity of the virus. In addition to targeting enzymes that are found in other organisms that are clinically relevant to humans, bacteria and viruses, we can also target human enzymes. And a number of the drugs which are in clinical practice for physiological problems within the human body are active against specific human enzymes. What I'll do over the next few slides is talk about a couple of examples of these. And what this goes back to is what I introduced at the beginning, that enzymes are involved in every single physiological function. They're involved in the rate at which we breathe. They're involved in our blood pressure. They're involved in brain function. So if your enzymes are going wrong, or if there's a physiological process which is not quite right, then enzymes are involved in regulating those processes. Now, if we can design and use drugs to alter that regulation and change the way in which those enzymes are functioning, it therefore is the case that you will alter the physiological function downstream of that enzyme. And because of that, you can use small molecule inhibitors targeting enzyme function to modulate human physiology. It's very much the case that a number of diseases are also caused by specific changes in enzymes. A number of cancers are caused by mutations in the human genome that alter enzymatic function. A number of the genes that I'm interested in my research, I work on Parkinson's disease. A number of the genes that are impacted in Parkinson's disease are enzymes. You have mutations in those enzymes, their function is altered, and therefore you want to try and find ways in which to change that back. And we can use drugs to target those changes. Well, the first example I'll run through is angiotensin converting enzyme. This is involved in regulating the function of the kidney, and it acts as a protease. So actually, in terms of its fundamental function, the class of enzyme that it belongs to, it's exactly the same actually as the HIV protease. It carries out the same task. It binds a peptide sequence and it cleaves that sequence, releasing substrates. And the specific reaction that it catalyzes is the cleavage of angiotensin 1, converting it into angiotensin 2, the active form of the peptide. And this has a number of physiological consequences in terms of the way in which our bodies function, one of which is to increase water retention. This is basically the role that angiotensin converting enzyme carries out. So you have angiotensinogen, which is a proprotein produced by the liver. This is converted by another protease called renin into angiotensin 1. And then angiotensin converting enzyme, released either by the lungs or by the kidneys, processes angiotensin 1 into angiotensin 2. It carries out a proteolytic event, binds angiotensin 1, cuts off a bit of it, and releases angiotensin 2. This then has actually quite a wide range of physiological impacts, altering the activity of the nervous system, altering the way in which our kidneys function in terms of tubular function, impacting on the release of aldosterone, which also impacts on kidney function, and impacting on the function of the arterioles, altering and directly impacting on blood pressure, as well as impacting on the pituitary gland and increasing the release of antidiuretic hormone. So it has quite a pleiotropic effect. It affects a lot of different things, but the key impact is that you have an increase in both water and salt retention. The more water you have in your body, the more will be in your bloodstream, the higher your blood pressure will be. So this is a summary of the enzymatic events that occur during this process. So you have angiotensinogen released by the liver. This is processed by renin into angiotensin 1. And actually what you can see here is the amino acid sequence of that peptide. So you have a sequence here with a much larger protein sequence towards the end, C-terminus. This is cleaved by renin to release a smaller peptide. You then have angiotensin converting enzyme cutting off a small sequence at the end of the C-terminal of the peptide, which is then processed into angiotensin 2. There's then further enzymatic events which reduce the size and activity of the peptide thereon. Now, clearly, blood pressure is a key physiological process which needs regulating. Many people live with high blood pressure, and this has potentially very serious consequences in terms of their day-to-day life and also in terms of their life expectancy. Blood pressure can result in a number of problems, both in terms of heart function, but also in terms of brain function. If you have high blood pressure, you're more likely to have a stroke, for example, and develop bleeds within the brain. And so clinically, we need ways to modulate blood pressure. One of the ways in which we do this is by targeting angiotensin converting enzyme. And a number of drugs, including ramipril, which is made by Sanofi, originally developed by Sanofi, target angiotensin converting enzyme and competitively inhibit the activity of angiotensin of this enzyme. And here again, you can see a crystal structure of angiotensin converting enzyme. And instead of the peptide that it's meant to be cleaving in the active site, this has ramipril bound. What it does is it decreases the activity of ACE, it reduces the amount of substrate, the amount of product which is produced, decreasing the production of angiotensin II, decreasing fluid retention, so more liquid is going into the urine, more liquid is coming out of the body, and blood pressure is thereby reduced, having a very important clinical benefit. And this is one of the key ways in which we can modulate blood pressure in using a drug. Final example I'll talk through in terms of modulating human physiological activity is cyclooxygenase. So cyclooxygenase is an enzyme which is responsible for the synthesis of prostaglandins. These are a critical component of our inflammatory response. So when our body responds to an outside threat or to an internal shock by generating an inflammatory response, cyclooxygenase is part of the cascade of events that allows our body to react. And cyclooxygenase inhibitors, abbreviated to COX, inhibitors can reduce the synthesis of prostaglandins by inhibiting the enzymes that produce prostaglandin. This is the chemical structure of prostaglandin shown down here at the bottom. In terms of the biosynthetic pathways that occur here, what you have is diacylglycerol or a phospholipid modified into arachidonic acid. This is then processed by cyclooxygenase 1 or 2 into the prostaglandins. These then undergo a series of further modifications, but this key step here is critical for the inflammatory response to occur. And actually, we have a very highly used way to modulate this in clinical practice. This is ibuprofen, which was originally developed by the Boots Company up in Nottingham during the 1960s and 70s. And what this does is it acts as an inhibitor of the cyclooxygenase enzymes. So what it does is it prevents the conversion of arachidonic acid into prostaglandins. And by modulating that step, by reducing the production of prostaglandins, you dampen the inflammatory response. And this is how ibuprofen mediates a clinical response in humans. So when you take an ibuprofen, what it's doing is inhibiting an enzyme, acting as a competitive inhibitor of that enzyme. For the last few slides, what I'll talk about are some of the other targets that are either emerging or now being used in clinical practice. And one of the key examples I'll use are protein kinases. This is an article by Philip Cohen, now Sir Philip Cohen, who leads a drug discovery institute at the University of Dundee, and who was a key player in understanding how we can target kinases in the context of human disease. And protein kinases are a very, very important regulatory element in the way in which our cells organize themselves and the way in which they direct their activity. They form a key part of signaling cascades within the cell. These are the events within the cell that allow a cell to respond to changes in the environment or alterations in the way in which the inside of the cell is functioning. And they carry out actually a fairly simple chemical reaction. They hydrolyze ATP, adenosine triphosphate, and they transfer the gamma phosphate of ATP, that's the final one in the three stuck onto it, onto other molecules. Sometimes, not always, but most frequently, to other proteins. By sticking a phosphate group onto another molecule, either another protein or other chemicals within the cell, you stick a very large charge group onto that molecule. By doing so, you alter the three-dimensional organization of that receiving substrate. This is a very simple cartoon explaining what kinases do. They take this is a blob representation of a protein kinase. It binds to ATP and hydrolyzes it into ADP, adenosine diphosphate, and a phosphate group, modifying either itself or other proteins within the cell. And so what the kinase does, it acts as a biological catalyst. It breaks up that phosphate bond there, releasing the phosphate group and sticking it onto other chemicals. And if you stick that highly negatively charged group onto another protein, those negative charges disrupt the way in which that protein folds. Those negative charges repel other negative charges or attract positive charges in the structure of that protein and alter the way in which that protein functions. Quite often, actually, a kinase will modify other kinases, creating a cascade of events that regulates processes such as transcription or the degradation of products within the cell through the lysosomal system. And actually, this is a key area of drug discovery, especially in the fields of oncology and increasingly in the field of neurodegeneration. This is BCR-ABL or c-ABL, a kinase which is involved in regulating cell division. If you have dysregulated cell division, one of the consequences of that can be the occurrence of cancer. Cells that are uncontrollably replicating within the body, forming tumours with potentially devastating clinical consequences. By modulating the activity of the ABL kinase domain here, you can alter the activity of the signaling cascade that regulates cell division and reduce cell division. And this is a drug called imatinib, which is used in clinical practice in oncology to prevent the uncontrolled replication of cells. This is a drug that was developed by Novartis and is used in chronic myelogenous leukemia, and is one of the ways in which we can target kinases within the cell that have gone wrong to modulate cell division and treat cancer. Final few slides, I'll talk about one of the other ways in which we can target enzymes. The inhibitors that I've talked to up until this point are inhibitors that directly target the active site of an enzyme, that pocket, the three-dimensional pocket on a protein which binds the substrate directly and actually is very specific for a certain set of substrates. But there are other ways in which you can modulate the activity of a protein or modulate the activity of an enzyme. And as well as binding directly to the active site, actually you can bind to other sites away from the active site that can alter the overall protein shape, the conformation of the protein, and that in turn can alter the way in which a protein and an enzyme interacts with its substrate binding and enzymatic activity. So going back to the enzyme kinetics, this is a plot showing the initial rate of reaction in the presence of increasing substrate concentration. This is for a standard enzymatic reaction. Where you have allosteric modulation of a reaction, what you get is a slightly different series rate of advance. So instead of having that exponential increase at the start, you actually have an S-shaped plot of the reaction. And this is because binding of chemicals to the enzyme actually alter the way in which that enzyme functions. So the events that allow an enzyme to function alter the shape of that enzyme, potentially make it more likely to bind more substrate. The classic example of this is the binding of oxygen to hemoglobin. And what you get with that, hemoglobin is a multi-protein complex. It has multiple constituent proteins within its quaternary structure. The binding of oxygen to one of those globin units actually alters the shape of the other globin units, making it more likely that they will bind oxygen. So what you get actually is this S-shaped curve that represents the initial binding of oxygen here, that increases the likelihood of binding other molecules of oxygen to the other subunits of the hemoglobin complex. And this is a classic example of an allosteric effect, one where binding of a chemical or a substrate to one area of a protein has an effect on the other parts of the shape of the other parts of the protein. And again, you can target this in drug discovery. So here we have a number of examples where you have an activating chemical which binds to an enzyme, unlocking the active site and allowing a substrate to bind. This is the normal situation where acting physiologically. Now, if you have an inhibitor that can bind to that activation site and prevent it being activated in the first place, actually what you can do is you can prevent the substrate binding at all. So binding away from the active site alters the shape of the active site, prevents the substrate binding, and therefore prevents the enzyme from working. You can also have a situation where, in a physiological situation, you have a molecule binding away from the active site that alters the shape of the active site in a way that allows the substrate to bind. And again, you can distort the active site, and this is shown here, where the active site has been distorted so the substrate can't bind. You can also alter the formation of a protein complex. So, for example, if a protein requires multiple individual proteins to interact to form an enzymatic complex, you can use an allosteric modulator to prevent dimerization or trimerization or tetramerization and prevent the formation of that enzymatic complex and prevent the enzyme functioning, prevent the enzyme developing an active site that can be used. Now, these are actually, although the concept behind these are quite simple, actually carrying out allosteric modulation of enzymatic function in clinical practice is much more difficult. The reason for that is that the active site of a protein is very, very specialized and designed to bind specific substrates. So if you can find something that is specific for that active site, you can get a very specific impact. You can target just that enzyme. Targeting allosteric regulation, where you're looking at sites on the outside of a protein or looking at protein-protein interactions at an interface, chemically and structurally is much, much more challenging. And doing so actually is it's much, much more difficult to avoid off-target effects, where you're targeting other proteins that you don't want to alter in the context of drug discovery. So there are actually very few examples of allosteric enzymatic regulators in clinical practice. There are examples where cell surface receptors can be modulated allosterically, or ion channels, but these are not directly targeting enzymatic activities. So just in summary, enzymes are proteins that carry out tasks within the cell. Hopefully you're all well aware of this, and that they can be described using enzyme kinetics. You can target them in a number of ways: competitive and non-competitive, irreversible and reversible. And these can be altered by direct binding to the active site or by allosteric mechanisms. I've put a lot of extra reading on the Blackboard pages. Feel free to read through those. You will not be examined on those, but they will give you a lot more information relating to what I've covered today. And of course, happy to answer any questions if you have any. Thanks very much.